This short video is to address a question about the confirmation or diagnostic PCR that was done in this practice exam. So I want you to look at the original plasmids that we are trying to differentiate between. We started out with P-Glow Topo here. And in addition, right, in order to make this new pecan MLOC wild type, I won't consider the delta 200 for this video, we had to amplify a region of DNA from some template, right, that was MLOC, so probably from the mouse genomic DNA. So both of these pieces of DNA, the MLOC and P-Topo, were put together so that MLOC could be inserted into the PCR bindings, into the PCR product site here to produce this new plasmid. And the question is now, do we have this plasmid? And that's what the diagnostic or confirmation PCR is set to address. Both of these plasmids, when transformed into competent cells, competent E. coli cells, would result in growth on LB plus AMP. So we can't use the phenotypes in this case to confirm that we indeed have made the right plasmid. So PCR is what we use. And as you see here, we have several potential primers that we could use. We have the original primers that were used to amplify MLOC, which are designated primer 3 and primer 4, right? That's what was used to amplify MLOC from the genomic DNA, and those are found here and here on the pecan MLOC wild-type plasmid. They are not, they will not recognize any sequence on the P-Topo plasmid because we haven't inserted that piece of DNA yet. Conversely, the primers 2 and 1 are recognizing sequences that are embedded in this original P-Topo plasmid. And although they're not listed here, because that was part of what we were assessing, primer 2 would recognize a sequence roughly here, and primer 1 would recognize a sequence roughly here, simply because we didn't have the P-Topo sequence itself was not modified, since we just added another piece of DNA into it. So now let's think about which of these primers we could use to confirm. We could use primers 3 and 4, right? But in this case, we would get product from here, so 1,300 base pairs here, and product if we still had this piece of DNA floating around. And so because we're not seeing the backbone of the P-Topo in the confirmation PCR, it's hard to distinguish between these two possibilities. Instead, if we use primer 2 and 1, then we would get a product from the original plasmid that would be different in size from the product from the pecan MLOC wild-type plasmid, the engineered plasmid. And that difference in size could both allow us to see that PCR is working with both templates and confirm that there is an added piece of DNA inserted, because we know what P3 and P4 were designed to amplify. We know exactly what size that sequence is, so we have very clear predictions about what the size of the products are going to be. And so that's why it is more appropriate to use external primers external to the PCR product that you've engineered into into a plasmid to do your confirmation or diagnostic PCR. We also now have a prediction about, depending on what size this product is from p-topo, so we could say X base pairs, right? Whatever is amplified from the pecan MLAC wild type would be 1300 plus those X base pairs, right? So then we can check on our confirmation PCR gel to see if that fits. So here we have our confirmation PCR gel. It looks like the band amplified from p-topo control sitting here is at 500 base pairs by reading off of the ladder, right? So now we can calculate that the expected product from pecan MLAC wild type would be 1800 base pairs. And that is consistent with what we observe on the gel.